hard break, bad DJing, but good morning. Good morning, good morning, good morning. Thank you for all of the artists who have adorned this ancient lithograph of calcareous sponges. Hello? Yeah?
It's the end of class.
Ok. Peux-tu demander à maman maintenant? Merci. Okay. So, welcome. Welcome to the Monday where we start diving into different groups within the Dun phylogeny. We're going to spend a lot of time with sponges today. We have a little bit of catch up to do from some of the activities we did at the end of last week on Friday. So, let's just jump in. And I wish to save that. Thank you for all the drawings. We're going to clear all those. And we're going to continue. Here we go. So, first thing I wanted to ask was whether or not you had a great weekend. This was some of ours. We spent it outside in the backyard, doing lots of skating and staying warm by the fire, and had some visitors. This is, of course, a Photoshop thing. We wouldn't have visitors. We're all being physically distant. First thing we're going to start off is a reminder that you'll see on CourseLink today that the first scavenger hunt the details are up online, and it's all about song lyrics. So you can read about it here, but two of the volunteers will be monitoring, Chantal and Jasmine will be monitoring this challenge, and so you need to find a song that has an invertebrate in the title or the song lyrics. There's a collaborative playlist on Spotify, but you don't need to put it there if you have some objection to Spotify. The critical thing, it's a collaborative playlist. But you need to be able to post it into the discussion forum about this first scavenger hunt. And another reminder down there at the bottom that if you haven't already done it, your personal introductions are due by the end of the day today. Hello, everyone just joining us. So there's a look at some of the Spotify lists that Sherry and Jenny and Carolyn and I have started to add some things to, and you're going to make it a diverse, super duper playlist of invertebrate musical genius. So today, where are we going? We're going to finish talking about the origin of metazoans, and we're going to start talking about the specifics of one taxon, one phylum in that Dun phylogeny, the Porifera, or the sponges. So for where does that mean in your text, you can see down there that chapter 4 and 5, these kind of overview chapters about introductions to life history, animal life histories, and body plans and architecture, which are chapters that we're going to refer to throughout the year. So these are kind of really key ones, to keep jumping back into and consulting. And then chapter 6, where Brusca and friends have lumped together two metazoan phyla, the Porifera and the Placozoa. So we're going to start talking about the sponges, the Porifera, today. Keep talking about them Wednesday, and then briefly introduce you to the Placozoans, where there are some pretty amazing things happening that we'll talk about on Wednesday. But I must not jump ahead of myself, because the first thing we have to do is finish up what we did on the last question that I asked you about on Friday. Bernie and I asked you to spend some time in the breakout rooms at the end of the class to talk about why that ant that was suspended, the weaver ant holding that little tiny mass, many thousands or hundreds times of her weight, why she was stronger, or why is the ant stronger than the person, how and why. So let's talk about that briefly here, because I think, if you see down there at the bottom, that other sheet, the advantages of multicellularity, you had a lot of ideas there, and you'll see as you go back and look at these lectures that most of your ideas are in the ones that I wanted to capture the subsequent slide later on. Why is the ant stronger than the person? How or why? So here, I think there's a couple of things. We introduced it at the end of class, so probably you were leaving already. But some of the ideas here, could have been stronger because of the exoskeleton, more legs, evening out the weight, more muscle compared to body weight, and they use their muscles for strength because they're so small. So the third one in there, more muscle compared to body weight, that's on the way to being the explanation as to why small things, many small invertebrates, are compared. You can see them wandering around carrying giant comparatively, relatively gigantic objects, and probably since you were a child lying on your stomach looking at ants carrying big things or other small invertebrates carrying things, and you're like, wow, wouldn't that be amazing if I could just kind of lift up the car and walk down the street? So why can't you? Excuse me. And it's because of this physical property that the surface area, this is a generalization and it kind of breaks down when we get into intraspecific examples in our own species, but the surface area of the muscle in cross-section is what roughly determines strength. So it's a surface area, but all else being equal, it's the volume of the animal that determines mass. So you can see one of the things we were touching on at the end of the week was relationships, the importance of relationships, squared and cubed relationships between surface area and volume. And this is one of them, and one perhaps that you haven't been introduced to in the past. So if we consider three theoretical cubes, so we've got, and these are our muscles that we've cross-sectioned from our square Lego person, for instance. So the strength to weight ratio, which will be the surface area to the volume. So if we have this 2 centimeter cube, the strength to weight ratio is proportional to about a half. So that's kind of, that's our small example. So think of this as the ant. And if we jump through these other examples as we get a larger cube, you can see that ratio changes very, very rapidly. And so the ant is stronger essentially because she's smaller and because of this relationship that strength is related to the cross-sectional area and mass is related to the cubed volume. So it's as simple as that. So you can wow people as you're sharing physical space within a vaccinated future when you're all lying on the grass this summer, late in the summer when we're all vaccinated and looking at ants and wondering why they can lift such large weights. You can say, well, actually, it's because of the cross-sectional area of the muscle. All right. So surface area to volume ratios, this is all about how to be a metazoan, remember, of lots and lots of different importances, and we're going to talk about these throughout the course. How can you get oxygen to internal cells? How to avoid cell waste? There's physical limitations. These physical limitations of surface area and volume, the relationship of squared to cubed, ignore direct possible evolutionary solutions. They're important to us because of how we respire, how we lose water, how we lose heat, strength, and responses through evolutionary time in multiple different lineages often involve changing internal surface area, changing microvilli, cilia, trachea, or the external morphology. So one example, a vertebrate example for ourselves, is that if you imagine a tennis court and you think about if we as humans had to respire through our skin, we might have 2 meters squared of skin to respire through. But the surface area of the lung, if you spread out all the alveoli, it's about the area of that tennis court. And so we've responded, so our response to the limited surface area that we have as a medium-sized large object is to increase the surface area of the respiratory surface internally. So this will bring up a couple of, so it's a vertebrate example, but it's you, it's important to you. Respiration is important to all of us. But I bring it up because it's a strategy that involves a couple of key aspects that are going to be important as we talk about the origin of metazoans, and that's differentiating internal from external and changing surface area by changing specialized portions of our internal anatomy. So what are some of the advantages of multicellularity? We talked about that last week, but here's a kind of a catch up from all of the things that we talked about, that we can divide labor across the body, that you can have specialized tissue leading to specialized organs. You can have efficiency. Some of you brought that up in terms of that you can lose part of your body without losing the whole. It allows an increase in size because we don't have to deal with just the simple rates of diffusion as far as limiting how far oxygen can penetrate or how far waste can be excreted. We can change different strategies. We can add complexity to how we live. We can have predators. We need to then evolve subsequent adaptations to prey or predator avoidance. So we have the initiation of this red queen of predator and prey kind of racing through evolutionary time. Complex tasks, information processing, and speed. Things get faster once you're a multicellular organism. So that's kind of the tail end of the trying to be a metazoan. And what I'd like to do before we go much further is just take a check-in to see if I'm missing anything in the chat. And super. There's some questions in there about how long the intros need to be, and the answer is about a minute. Do you need to post the scavenger? If you can, that's exactly. Oh, hello, Sherry. And I think everybody's up to speed there. So we will leave this. We have verified the shot, and we keep going. So, metazoans. What are we talking about with metazoans? Now, in your text, as I'm sure you've all seen, there are two simple definitions of protozoans and metazoans. Protozoans, Bruska says, are eukaryotic, largely unicellular organisms that don't undergo tissue formation through a process of embryological layering. Metazoans are heterotrophic, multicellular eukaryotes that undergo embryogenesis by way of tissue layering. Okay, so those are two kind of simple definitions with some page references that you can consult as you go on. Now, we spend a lot of time talking about the diversity of protozoans, these eukaryotic but largely unicellular organisms, and we're going to spend the rest of the term talking about metazoans. And so one of the ways that we're going to start out is by asking ourselves about what hypotheses have we about the origin of metazoans. Now, Ernst Haeckel, who is a biologist in the 1800s, who is actually responsible for any of the colorings that we've done at the start of the lecture, those old lithographs, they're done by Ernst Haeckel in a giant tome of a book that he called Art in Biology. And it's a gorgeous book, and I can post the link later on if you're interested, because the full text of this giant tome is publicly available. That's where I'm getting them from, on the Biodiversity Heritage Library. So, as a thinker, one of the things that he was responsible for was the first kind of elucidation of the colonial theory of metazoan evolution. So the nub of this idea in the colonial theory was, you can see demonstrated on the right-hand side by a contemporary multicellular choanoflagellate. There's a particular species of choanoflagellate, the Protospongia haeckeli, named after Haeckel. Ta-da! Connections all around. So, his hypothesis was that multicellular metazoans derived from colonies of flagellated protozoans. And these flagellated protozoans, like these choanocytes that can live individually, but can also form groups and form these little balls that kind of go through together with all their collared flagella beating out to the external surface. This is an idea that he thought had support because he could imagine how you could have cell division without separation. Now, one of the competing hypotheses for a long period of time was the syncytial theory, where there was the metazoan was derived from something like a ciliated protozoan. Remember the micro and macronucleus bearing ciliate or ciliophorid? So a ciliate protozoan that had large multinucleate cells that eventually partitioned into compartments. Now, this has not had a lot of theory, and you'll see it raised and dismissed in your text quite quickly. And the reasons for that are that we can imagine many more advantages to the colonial theory that largely have to do with the separation of, how the conflict between locomotion and growth is resolved. That we've seen kind of contemporary examples multiple independent times, animals, fungi, bioalgae, but if you're going to divide, the problem with the syncytial theory is that you're going to not be able to continue, and if you're going to divide and reproduce, you're not going to be able to keep moving. Whereas in this colonial theory, that is facilitated. The other reason that we're attracted to it is because we see, and we have multiple lines of evidence through time that support the hypothesis that choanoflagellates are the sister taxa for metazoans. So it kind of puts it out there as being, okay, well, if we imagine that an individual choanoflagellate could become some proto-metazoan, it supports that hypothesis. Couple of people, welcome. So, how would this happen? You would enlarge existing cells, the flagella would regress, you'd divide, and then reassemble your flagella. So some cells can divide while others don't, and that division allows you a division of labor. So localized cells set aside for growth. So the criticisms of the syncytial theory is that it would limit body size and compromise locomotion, whereas the colonial theory, the CT, is that it would permit both, and it's a good, efficient compromise between growth and locomotion. So that's kind of the contemporary thoughts that we have about the evolution of a metazoan ancestor. Now, I mentioned when we're talking about surface area to volume ratios, one of the things that the importance to that is differentiating interior from exterior in a multicellular heterotroph. And so the epithelium, this part of the body that does that key differentiation between interior and exterior, is a key innovation that leads to the diversification of the metazoa. So as we look at this cross section, of the epithelium, you can see the base with the basal lamina and the apex. So the interior down here near the word base and the apex. So this allows, so it essentially polarizes the body so that we have basal layer facing other cells, facing the interior, and the apex facing out into the environment. And you can see even here in this example, the wavy, potentially cilia or potential villi, microvilli. Maybe this is something that's another attempt to increase the surface area to increase the rate of reaction. It also permits that continuous layer on the inside, the basal lamina, that then provides a clear differentiation between inside and outside, and also allows intercellular connections. And so these are a key step in the evolution of muscle and of nerves. There is the basal lamina there. So quickly talking about some key innovations in the evolution. I'm jumping back to the chat, and I see lots more people joining. Hello to you all. And I see no specific questions, so everything's chill and cool for the end of our discussion about the evolution of metazoans. And now, hooray, we enter into the Dun phylogeny with the first taxon, the first phylum that we're going to talk about. And these are the sponges, the Porifera, about 8,000, at least 8,000 species. So the Dun phylogeny, remember, this is our kind of the tree, the phylogenetic tree, the best hypothesis that we have for the evolution of our metazoan animal neighbors and co-inhabitants of this planet Earth. It's a slightly different version, you'll note, than what you see in your text. Now, we're going to come to that in a second. But remember, 32 tips here, lots of complex information, but we can simplify it across the nodes where there's lots and lots of shared information that supports the collapsing of those nodes, wherein we might not know how the platyhelminthes worms are related to the gastrotrichs within the spiralians, yada yada yada. We'll come to that. However, we're confident in the phylum of the taxon of the clade of the ecdysozoans and the spiralians, forming the protostomes, the deuterostomes, our own neighborhood, all of us together being the bilaterian, and then these amazing, principally marine groups where there is not bilateral symmetry, where there's radial symmetry, or where symmetry is absent. And so these are the first taxa that we're going to walk across as we climb through this tree. So the ctenophores, the sponges, Cellujac, placozoans, cnidarians. So here's the first example where the Dun phylogeny is slightly different than the phylogeny that you've seen in your text. So the poriferans, the sponges, are up here right at the top. This is a contentious phylogenetic statement about which there is not 100% agreement. Now remember, phylogenies are hypotheses. There does not need to be 100% agreement. There needs to be new evidence and new testing of those hypotheses. So what is the contrasting hypothesis? This is from your text, and this is essentially showing the position of the poriferans. So here you can see it's second in most basal. However, in this phylogeny, it is most basal to the choanoflagellates. And you can see where they have listed ctenophores in Brusca is listed with a dot. And that's referring to the contention that we're going to break down and talk about the evidence that supports the Dun phylogeny. And that's the one that I'm going to, and Cherry and I are going to keep talking about throughout this term, that ctenophores are kind of the sister taxa to the remainder of the animal groups. In talking about the sponges, we will talk about why, for example, I was taught that choanoflagellates, sister taxa to all animals, sponges, sister taxa to all the remainder of the animals, and it essentially has to do with the similarity of one of the cellular types of the sponge to the choanoflagellates. So this is a choanoflagellate. So remember that unicellular organism with the collar up and the beating flagella that filter feeds in the water, can occasionally group together and form complex, almost like theoretical hypothetical proto-metazoans? Well, there's a cellular type within the sponges that looks so incredibly like a choanoflagellate that we're left for many years hypothesizing that the sponges were then the sister taxa to all the remaining animal groups. So as we unpack sponges and ctenophores over the next say 4 or 5 lectures, we'll touch on those things. I'll unpack why we no longer think that. And so it's essentially a discussion about whether the morphology of a choanocyte in a sponge or a choanoflagellate are homologous. So what are sponges? Sponges are in the 5th chapter. The 5th chapter, 6th chapter. You'll see that sponge the porifera comes from Latin for pore bearing. And so the apomorphies that characterize this group include these choanocytes, where there are microvilli collars surrounding a beating flagella that create a feeding current in the living sponge. These arise from a single cell or almost a totipotent cell, a syncytial cell. And the original hypothesis was that these were homologous with choanoflagellates, the sister group for all remaining animals. The adults are sessile filter feeders. The larval stages can move about in the water. They're built, remember Attenborough talking about collagen as one of the structural most important proteins to multicellular life. They're built, but they can also build skeletal elements involving calcium carbonate, silicon dioxide, and collagen. So different components from seawater as well as proteins. And they create this aquiferous system that we will talk about in some detail today. So here is the phylogeny from figure 6.27 from your text, which is a look at the principal 4 lineages of the sponges. So we have calcareous sponges, we have demnosponges. If you were wondering where SpongeBob fits, he fits in here. I'm busy. And we have glass sponges. We'll talk about some of these phylogenetic hypotheses today, but first we're going to talk about some of how I mentioned the syncytial, almost totipotent nature of some sponge cells, and we're going to share another part of this BBC First Life video where we push sponges through a syringe and see what happens. Don't try this. Those of you who are joining us asynchronously, they aren't together right now, so you're not seeing that. What you'll find is that some of these videos that are proprietary videos in this example of the BBC, when I post the videos later on, you're going to have to go and look at that video on your own because I need to clip it and trim it from our video in order that we can post these videos and have you take a look at them. So in the chat, I always post the links. In the PowerPoint that turn into PDF that you all have access to, the links are always there. And so if you're looking at these asynchronously, these small videos are trimmed, and this part should still be there, me explaining to you how to look at that. So let's take a look at some of these cellular types. This is a cross section. This is a figure, so figure 6.8, cell lines cells that line sponge surfaces. 6.8 is not quite as good as this one from Rupert, Fox, and Barnes, but this is the equivalent one in your text. So there's a lot of different cellular types as you look at this vertical cross section of a sponge wall. And let's work our way from the top. Let's not work our way from the top down. Let's work our way to the one that I've been talking about, the choanocyte. This collared flagella-bearing cell that beats and creates suction inside the cell and then water flow from through the ostium from the exterior to the interior. And then the flagella captures little particles of food, moves them down, and then allows the sharing of that nutrition across all these different cellular types. So those are important that's an apomorphy for the sponges. These sclerocytes are another characteristic. This is a intracellular cell type that secretes either the protein or the calcium carbonate or the silicon dioxide. I scratched your head. I scratched your head.
Hey,
Yep.
Papa.
Yep.
I need some pain.
Okay. Peux-tu demander à maman, s'il te plaît?
Normal pain.
Merci, Jack. So that's the sclerocyte that secretes the spicules. We'll talk about that at a future time. The lophocyte is another one that secretes the collagen. The pinocyte, which is the squamous cell that creates the lining of exterior of the cell wall. The archocyte, these are the totipotent ones, the syncytial ones that become so important in that experiment of pushing a cell or pushing a sponge through a syringe. This may be the only taxon that we will do that experiment with in this class. And then finally, the porocyte, and that forms the ostia. The ostia is the critical connection through the side of the sponge from the exterior to the interior that allows water to flow through, containing little tiny food particles that the sponge will then feed upon. So there's suspension feeders that essentially, so this is again from Rupert, Fox, and Barnes, and so take a look here at this one that we have, that we flipped the orientation unfortunately on this, but we have water coming in bearing different food particles that as the choanocyte in the center, in that little choanocyte chamber, as it beats here, it creates the difference in pressure, draws water in, and tiny archocytes phagocytose and draw in those little tiny micronutrients and then digestion is completed inside the mesohyle and shared amongst all of the cellular body. So intracellular digestion. Now this design of sponges we have three different kind of terms, and if you go out to St. Andrews and you take the marine course and you take a look at sponges, you might describe them according to these three different terms that essentially have to do with how complex they look. So an asconoid sponge is a very simple one and tends to be very small. A synconoid is a slightly more complex, and you can see in red and yellow that there's more folds, interior folds in that wall. And then a leuconoid is the most complex, big, giant sponge with a whole bunch of different choanocytic chambers. So there they are, kind of lined up 1, 2, 3. Now we're showing them at the same size here, but remember one of the things that's changing from left to right on your screen is that from asconoid to leuconoid with increasing complexity comes the capacity for increasing size. But one important thing to remember is that this is not a phylogenetic classification. These are essentially words that describe how complicated they appear to be. Now, one of the reasons that we don't see large asconoid sponges is shown here, and it's essentially because as you get bigger and bigger, just with the chamber design or the chimney design of the asconoid, the top simple sponge, you see a large stagnant zone where water's not moving. And sponges are all about moving water. We're going to spend some time talking about that in 2 seconds once I jump over to vérifier l'achat. And I see no questions. So everybody's enjoying our meandering through the sponge community, or you're all sitting there with your coffee going, oh Monday. Oh, Monday. All right. So as we think about our sponges and how we move from asconoid to leuconoid, we think about these ostia, these pores that go from the interior to the exterior. So singular ostium, plural ostia, and the creation of these choanocytic chambers that create, the two of them, excurrent canals. And so let's take a look at what that results in is that sponges tend to have continuous flow and a velocity that's determined by the area, so you can have a greater velocity the greater the size you have. And before we break down what those choanocytic chambers can do, I have a short video here to show you. Oh, I was hoping to mute that because it's Monday and there's no like pan flute. We don't need it. So a reminder, if you're watching this asynchronously, go and take a look at the video later on. Now, so we're scuba diving. We've got a syringe filled with a colored dye. Inject it at the outside of the sponge. Remember, those are where the ostia, the dermal pores, are. What you're going to see is that this dye, what's going to happen if there's current, the dye is going to disappear towards the center, and then, poof, just like your chimney, if you had a wood stove, or if you've had a cabin or a cottage where you've got a wood stove, that you're seeing kind of the same principles, some of the same principles apply here that begin to act to draw that water up. We'll talk about that in detail in a second, but what are the choanocytes? So remember the choanocytic chambers here. This is another figure that I've lifted from Rupert, Fox, and Barnes, so you're not going to see it in Brusca, but you've seen it in these lectures and you've seen it in the PDFs that you have. So a sponge can pump enormous volumes of its body, its entire volume, in approximately 5 seconds, and that tends to be continuous. Now what we have here in these, so the diagram, the lines are going from left to right here, is that we have through the ostia, we have water, a current passing by at the top or the side. But interior to the cell wall, or the sponge wall, we have these choanocytic chambers where you have to imagine all those flagella beating. That's creating a difference in pressure, a reduction in pressure, and that's going to draw water in and across. So as you go across the cell wall of the sponge here, you can see on the top red line that as you go across the area increases where inside of those choanocytic chambers. So there's an increase in area to allow for the beating of the flagella, and because of that, there's these areas of high velocity near the entry and exit points, and then a slowing in the choanocytic chamber that allows feeding to take place. Now, what I'd like to do today is to talk a little bit more actively in a second about what other physical properties of sponges allow them to feed as efficiently, to be some of the most efficient filter feeders in the animal kingdom. And so I'm going to present this simplified view of a sponge here and talk about Bernoulli. Now, you remember Bernoulli, right? I see nodding across, and we'll check the... So in fluid or hydrodynamics, so as there is a non-conducting, as there is an increase in speed of the medium, so in this case water, but in your chimney outside of your house it would be air, there's a decrease in the pressure, and so that is filled in through an opening at the other end to fill in that subsequently low pressure. So this principle on its own was hypothesized. So remember, when we're thinking about sponges evolutionarily and morphologically and systematically, we've been really drawn into the choanocytes because of how they may describe how the sponges fit inside of the animal kingdom, but also in terms of how they feed. It was the first cellular type that we talked about today. And I've talked about how those beaten choanocyte chambers set up the pressure change that allows feeding currents to be established. Now, a long time ago, there was a scientist named Vogel, I think it was Simon or Steve Vogel, that published a study that we're going to try and reproduce together to theoretically, conceptually reproduce the results of today, where he was convinced that choanocyte beating was not the entire thing that created the beating or the feeding current, the in current of the sponge. And so he was convinced that the Bernoulli principle was involved in the establishment of feeding currents by sponges. So Vogel did this experiment where he created zombie sponges, where he killed and immobilized the choanocyte. So half of the experiment was live sponges, half of the experiment were these zombie sponges where he had killed the choanocyte cells, and then he compared across different flow rates the flow rate for the zombie sponges and the living sponges. And so what I'd like to do is to ask for your graphical prediction of what he found for those two categories as the flow increased. So what I'd ask you to do is take your zoom annotation tools, and if you can, show me zombie sponges in red and live sponges in black, and show me with lines what your prediction would be if Bernoulli is involved. So as the external current on the x-axis increases, what should we see in the oscular flow rate? Remember, that's the connection, that's the current, the feeding canal across the wall of the sponge. So a live sponge in black, what should we see if Bernoulli? So if there's an increasingly fast flow across the top. And what should we see in the dead sponges? I'll give you a couple of seconds to annotate and I will drink some tea. Oh, interesting. Oh, we have two hypotheses, two groups. So I see lots of hypotheses in red for what the zombie sponges, and what are the live? Yes, now I'm starting to see some live sponges in black. I like this. I have to remember I want to save this. Awesome. Well done. Okay. Saved again. Now, so stop sheath your pens. I am going to clear all these drawings. They're gorgeous. But let's talk about, before I remove them, let's talk about what your hypotheses were. Maybe I can jump to the chat and try and do this coincidentally. So, hypotheses about what your zombie sponges should be doing there, if there's a Bernoulli effect on the feeding flow rate. So what you've shown me in red is that you would think that they would continue to increase. Now, one of the things, I'm going to jump out of the chat, come back to the slides, clear your gorgeous predictions, and I'm going to show you what they found. So this is the results that was published in the Proceeding of the National Academies in 1977. PNAS is about the third biggest journal in general science. And what they found on top was, in red here, was the normal flow. This is where we would expect to see, these were the living, so choanocytes creating these feeding currents, and then also the addition of Bernoulli. So occurring that positive relationship, so positively affected by the current on the x-axis, that's one key point, but it's boosted up onto the y-axis. Now, what he saw and what he used to interpret the fact that Bernoulli alone creates some of these feeding currents is the fact that when you kill the choanocytes, the relationship down there on the bottom, it's still here. It's still positive. It's just lower down. Does that make sense? So this was the work that was used to demonstrate that both Bernoulli and the choanocyte beating are what's contributing to the creation of the feeding current in the sponge. I love it. Except for the killing of the choanocytes, I suppose. That makes me sad. Okay, so let's jump back over to the chat for 1 second. Yep, we can talk about that, but it is not possible. I've talked about this several times. It is not possible to put the slides up because the slides are being made right up until the moment that the Zoom is opened. And so the point is not for you to keep up, right? You get the slides. I'm not breaking down the slides and producing only a subset or a bulleted list. You're getting all of these slides within the hour after the lecture is done. So the point by not producing the slides is A, dealing with the fact that physically I can't do it, and B, it's actually pedagogically it's better for you because you need to be, the point is not to have you writing down everything that I'm saying. The point is to have you try and distill and have your brain create the links to tell you what are the key points of what it is that I'm saying. And then if you want or have time, you can go back to the lecture and slow it down or watch me at 2.5 speed later on, and or just consult the slides in the next hour. But it's a critically important part of learning to have you distill and break those down. So, yes. All right. Now, what we're going to do here is pause for something a little bit different in the last 10 minutes. We're going to jump from talking about sponges, and I'm going to post a pre-lecture kind of 5, and it's going to be 5 or 10 minutes about sponges, but I'll do that this afternoon or tomorrow. But I want to set you up a little bit for some of what you're going to be doing in the lab today, and that has to do with not sponges, but these amazing creatures. This is where your nasal strip that came in your package comes into play. This is Demodex folliculorum. This is one of the 2 species of mite that lives on all of us, and we're going to try and sample and take a look at together. So this is a scanning electron microscope, classic one on the left-hand side from the interpipes. This is a particular chunk of my skin last year where I was elated to find some of my own facial mites because in previous years I hadn't found any, and that made me very sad. This is a look in video at the right-hand side of this kind of bicycling legs of this mite that's living in a facial pore, as well as an SEM of a Demodex, I think from your TA Jenny Gleason, that was taken by Mohammed El-Mankabadi, who was a student in this class and also a student in my lab. So the next 10 minutes we're going to talk a little bit about the background about Demodex, why we think it's interesting as a project that we can work at together at a distance, what kind of questions we're going to ask. So there's lots of detail here, but you can sit back and relax and just let this wash over you. All these links are going to be posted in the chat in a second, and you're going to be talking about this in your lab period this week. So we've known about Demodex for a long time. Like even back into different Aztec writings in 1552, probably we've seen some definition and description of what Demodex looked like. And that's because sometimes this naturally occurring mite can have an outbreak, and a large outbreak that's associated with rosacea and some symptoms on the skin. In Europe, it was first detected on corpses in the 1840s. The breakdown of the word, it means a fat worm, demodex in Greek, and folliculorum just refers to that in Latin that it occurs in the follicles, hair follicles. And so over the next kind of 50 years after its first description in the 1840s, people found them and discovered and rediscovered them. One of the macabre things is that they were found to be alive and viable on corpses for up to a week after the human had died. And up until 1972, which as someone who was born in that year, I don't think that's a long time ago, but it's half a century ago almost, we had one species until in 72, one was described, there was a split described, and that's what you're seeing here. Between the left-hand side, the longer Demodex folliculorum and Demodex brevis, or the brief or short Demodex. We're going to talk about, in the next few minutes, about the two of these species and where they live, some of their biology. So here's a cross-section, kind of a textbook-style cross-section of a hair follicle. And in blue, I'll draw attention to the folliculorum there at the base of the hair follicle. And then in the sebaceous cyst over here, the shorter brevis. So this is a deep genetic divergence between these two species. So we don't know a lot about this group. What we're still learning lots of important things about them. So only 50 years ago, we broke them up into two species. But we date the split, the evolutionary split of those two lineages, to be before the radiation of placental mammals. So this is a long time ago. And they're considered, this is a super important part. I'm hoping inevitably some of you are kind of, your skin is crawling as you learn about these invertebrates that share and call you home, but they are a completely normal part of the human skin fauna. Folliculorum, the larger of the two, predominantly lives on the face and lives around the hair follicles and is clustered around the follicle base. So in a cross-section of skin from deep to the surface, folliculorum is found closer to the top. Brevis tends to be found more solitarily and lives in sebaceous glands, and so it can be found kind of anywhere, and you might find it more frequently on the trunk or in your armpits, those kinds of things. You can find them in the same place, and you can even find them in the same gland, and so you might find them with the same kinds of sampling. So there's a look at, so that jumping away, so this is now a cross-section of a skin biopsy, and you can see these kind of long elongate forms. These are Demodex folliculorum. And there's a brevis up there on the top. So again, brevis, the short one, folliculorum, the longer one. So these are two species that you may have living on you right now, and we're going to try and sample together. So folliculorum can cohabit inside of a hair follicle, and I love this kind of colorized SEM of the tree trunk of the hair follicle and all of these little folliculorum butts. Now, how are they transmitted? They're transmitted shortly after birth, we think. And it's thought to occur from mother to infant transmission that occurs during nursing. And if you were to sample yourself for folliculorum through your life, the abundance of them would be kind of low after that inoculation from your mother. And then it would tend to spike around puberty and increase for the rest of the time that you live. And that has to do with abundant food supplies that are associated with the glorious bits of food that you produce in your sebaceous cysts as you go through puberty. 95%, it seems like in people older than 70 in this particular species, older than 71, very specific, individuals have Demodex folliculorum or brevis. And they're spread easily by skin-to-skin contact. And so people who work in long-term care facilities tend to have a lot of them because they're shared with those that they are caring for. Here's another colorized SEM of that folliculorum cluster at the base of the hair cell. So they're consuming cells. The Demodex brevis are consuming byproducts of the sebaceous gland. They move very slowly. They seem to be negatively phototactic, so they respond negatively to light, so they tend to come out or closer to the surface at night. They have a low tolerance for heat and dryness. And so one of the neat things about this is the adults lack an anus. So they're feeding and feeding and feeding on your face, but they're not really pooping. They just kind of die as a poop-filled organism. And the entire life cycle takes about 15 days, all on your face. One of the neat things that we discovered a few years ago, and there's going to be, I'll put some videos up on CourseLink or on the chat here in a second, and you'll see them at the end of this PowerPoint, is that different researchers started to sample genetic lineages of people for their Demodex folliculorum, and what they found was multiple deep lineages of folliculorum that were broadly consistent with where their parents, where the host parents had come from. And this was interpreted to support the hypothesis that the evolution of the mite reflects ancient human population divergences. And so we see deep lineages across North and South America, Europe, Asia, and Africa. And so what are we going to do as we sample these different Demodex mites from your face this afternoon, tonight, and tomorrow, and then pop them in the mail? So what we're going to try and do with this project that we're all going to participate and share together is to ask questions about how many mites will we find across the class. If we have roughly 200 people, are we going to find 200 mites? Are we going to find 50 mites? What's the range and the number of mites that we can find for host? As I mentioned, I've been in years past, I've been a 0, and I've also had up to 5. We're also going to ask you to fill in some standardized information about your parents' ancestry, a simplified version of sex, if you're comfortable sharing that, whether or not you live with pets, and whether or not you moisturize, so we can make some correlates about how many mites we're finding with maybe how dry or not dry your skin is. We're going to try and sequence these mites. We're going to sample. You're going to mail them back to us, and we're going to extract DNA from those samples where we find mites, and then sequence the mitochondrial region of cytochrome C oxidase gene that is used across animals as a DNA barcoding locus. And we're going to look at the genetic variability that's evident in the mites that we find. And then we're going to ask the question about whether that genetic variability is associated with ancestry. So, tonight, in the next few nights, what we're going to ask you to do is to take your nasal strip, open it up, sleep with it on your face. You can choose like a nice kind of grainy part of your nose. You can choose your forehead. You can put it anywhere. Record that information, share it with us. The strips don't contain latex. Remove it, place it back in that wax paper slip, and fill out your data form. That's critical. And then on your consent form, put them in your self-addressed stamped envelope and ship them back to us. So Sherry and team are going to be talking about this with you again in the specifics of how to do it this afternoon and the next couple of days when you have your labs. And then it'll take a little bit to have to do the sampling and have the sequences created, and then we'll rejoin later in the term after reading break, and we'll revisit this and test some of these hypotheses together. This is we're excited about this. We're excited about this science that we're doing together in a physically distant manner. So I'm going to take a second, and I'm seeing Sherry reiterate here, don't use the strips until you've had the lab. So if your lab is Monday, don't do it. You might do it tonight, but if your lab is not till Wednesday, wait until you've had your lab. Then get those strips back to us. So I'm going to shut down the Zoom in a second. I'm going to stay on the chat. I'm going to post a bunch of links, and I'm going to say have a great middle of the week, start of the week. We'll see you on Wednesday, and enjoy your labs this afternoon. I'm very excited for face mites. I hope you are too. Have a great Monday, and we'll see you soon.